The topic of this webinar is a sensitive and simple STEC solution. My name is Felix Hesler. I'm the product manager for the microbiological portfolio of Eurofins GeneScan technologies, working mainly with PCR and ELISA methods. I have been with the company for the past 4 years, and before that have worked altogether now almost 10 years in the food safety environment, and mainly working with molecular methods for pathogen detection. I was previously at Thermo Fisher Scientific working with these solutions there. When we start with STEC, of course, I think I would like to give a short overview at the beginning, introduce reference methods or other one reference method in this case, and some regulatory aspects, then go into the portfolio that we have for the STEC testing and show you some special features and introduce the workflows. Then I would like to highlight some parts of the certifications, and at the end of the presentation, make a small comparison and maybe also say what we think is the key features and competitive advantages of our methods. And then, as I mentioned before, go into the questions and answers. So I know this will probably be familiar to most of you, but when we talk about STEC, I think we do need to go a little bit into the background as well, because this does have an effect on lots of the regulations and these topics. So I think everyone is aware that Escherichia coli, the gram-negative from the Enterobacteriaceae, coming from a group where we are well familiar with other pathogens like the Salmonella. However, this one is a rather common bacteria, and most of them are non-pathogenic. So the question is, what makes some of them pathogenic? These pathogenic ones are the STEC, or the shiga-toxin producing E. coli. They can lead to bloody diarrhea, cramps, vomiting, typical symptoms, but also quite severe diseases such as the Haas syndrome, which I'm sure most of you have heard of and are familiar with, which is a very severe and in many cases lethal disease. So another aspect is the very low infectious dose of these bacteria. So we really want to make sure that you don't have them in your products. But what are they actually? How are they defined? We have said that it's shiga-toxin producing, but there are lots of different aspects that different regulations look at. So the most common thing to look at is the serogroups of the E. coli, because there is a strong correlation between the serogroups and the association to diseases. However, this is very, very controversial. So while there is a good correlation, there is still no guarantee that not other O groups will be problematic. So this is really, if you look from country to country, viewed very differently. The most common point to look at is the shiga-toxin production. And this, of course, makes sense because this is very often required to cause a disease. There are two different variants. There's the STX1, which is less associated with human diseases, and there is the STX2, which is more often associated with also very strong and severe diseases. And then there are lots of other factors. It's actually a very long list of other factors that can have an influence on the diseases. The most prominent and commonly known factor is the intimine, coded by the EAE gene. This is an attachment factor that helps the cells, the bacteria attached to the cells. But this is not always required. So there are very prominent cases of outbreaks where actually there was no EAE involved. Still, it is considered one of the key factors. Then there are other factors that might get into the place of EAE, which are AGGR, AAIEC, and there are other factors that are associated with an increased risk. But in the terms of this presentation, and as most methods look at it, it's really mainly the STX and the EAE as virulence factors. Now, since we're talking about Australia, I wanted to start when we look at the regulation with a little bit of an overview on what it is that is done in Australia at the moment. There is a very strong focus on the export, of course, so it's very much dictated a little bit, I would say, from what other countries view in terms of the STEC. So basically looking at the history, I think the main market is the US, where they were actually the first ones to declare E. coli as an adulterant, or certain E. coli as an adulterant, with the 0157H7, and they have changed this regulation or this view in 2012. The FSIS declared also other 6 serotypes as adulterants, and we look into a little bit more detail on the next slide on how this is currently stated. Another market would be Canada, where they also have controls for the 0157, and I think probably the key factor for the US and the Canadian market is that they require the pre-export testing. So basically testing before the exporting, and then have in addition the port of entry controls. There are other markets that are relevant as well, such as the EU and Japan, for example, and these have STEC port of entry testing, but do not require the pre-export testing. So in Australia, you would need to adapt to all these different requirements then, of course, and have methods in place that are equivalent to meet basically these requirements. And this is then, of course, as you probably all well know, done by the DAWE now, who approve the methods and also do the auditing and of the laboratories that do the testing. So if we look at the different statements from the regulatory bodies, I wanted to take 2 examples because they're very much on the opposite poles if you look at it from the definition point of view, what an STEC is. One is the US situation. We have already mentioned that on the slide before, but if we look a little bit more into the details of what it actually says, the definition is quite narrow when it comes to the STEC. It's the 0157H7 that is declared as an adulterant, as well as 6 non-0157 STECs, which have an STX gene and an EAE gene. Sorry. So both of these genes need to be present, as well as the serotype. If we look into the definition of the EU, it's basically stating, well, the serogroups are not relevant at all, and presence of intermin gene is not relevant as well for a severe illness. This has been shown especially in Germany in an outbreak in 2011. I think this is something that has defined this statement from the EFSA very much, where an 0104 outbreak led to dozens of deaths in Germany and in other countries in Europe. And this 0104 also did not include an EAE. So I think it's based on a quite real scenario. However, when we look at it, this actually never really went into any regulatory requirements for the meat industry. So there are some in the relevant European norm 2073, but this is only for sprouts, which were in this outbreak the food that was associated with it. So why then, and on which grounds do Europeans then do the border control? If you look at the RASFF reports in Europe, you can see what kind of border rejections were done, and you can see that there are actually border rejections and quite a few based on Shiga toxin producing E. coli. And they come from all different countries. So what is this based on? This is normally based on on local or national guidelines. I take this example here from Germany, which is the DGHM values, risk values or warning values. And in this guideline, which is not a regulatory instrument really, so it's not, you know, it's not binding by law, but it is a guideline that is kind of accepted as law within Germany. Here, the Shiga toxin producing E. coli are defined as an adulterant in beef. So on this basis, then the regulatory bodies basically do the testing for the border control. Now, if we look at the reference methods, I wanted to look only at the MLG, so the US methods. Some of you are probably familiar with it. The ISO method, which is more relevant for Europe, is very similar to it. So we have the enrichment. In this case, it's in MTSB. Of your food product, then you do a first screen. In this case, actually, there is a commercial product in the MLG, which is the IQ check. So you can see this in the blue boxes, and to the left of it, in the orange boxes, you can see the products from our side, which are basically the equivalent. So we will go into the details of these products later in the presentation. So if the first screening check with the STX and EAE is negative, then you will go into a serotyping. You will run more assays defining which serogroup is in that. If you don't have any of the relevant one, you would also call it negative. So this is the difference to Europe. In Europe, of course, you would say STX positive, and then you have a presumptive straight away. So after this, then you will go into isolation. I think most people probably don't do the isolation, but then send it off to expert labs. But still, I think also for our workflow, this next part is still pretty relevant because I think it's actually quite nice tool. We do have IMS beads, so beads to get your bacteria out of the enrichment broth in our portfolio as well. So this is something that we can use later on. I will explain also as a tool to control if you have a true positive or not. In the MLG, however, you will go via these IMS beads that take your bacteria out of the enrichment by immunomagnetic separation. You will go onto plates. There are different treatments on the plates, like this acid treatment to reduce the background flora. Once you have done this, you then go into a further identification. So you will take latex agglutination tests and see if you can see any agglutination with your bacteria in question with the antisera. And also for this, Eurofins Technologies has a product, and this one is actually part of the MLG 5C. So it is mentioned to be used in the MLG, which are the Abraxas latex agglutination kits, which I will also introduce briefly later in the presentation. So once this has been done, you are quite sure that you have something, but you will then go into another subculture, and from these isolated colonies, you will then try to confirm, basically by PCR and other methods like the VTEC, the identity of your isolate. But as I said, this is a workflow that up to the end, probably only a handful of labs will do in each country. So now looking at the products and the features of our portfolio, I've introduced the kits already a little bit in the overview, but let's see what the key characteristics are. I think one of the strong parts of this workflow is that it's a modular workflow, so it's quite flexible for all the regulatory requirements that are currently in place globally. We try to be as efficient as possible, so we're not trying to use any antibiotics in the enrichment media. There is a very short enrichment time, but also quite flexible here. So you will see later that you can also enrich very long. So we try to make sure that we are as flexible as possible, depending on what your needs are in the lab and basically from how the samples come in. And of course, since we do need high volumes of samples, the sample to media ratio is something quite important. So we try to keep it as low as possible. We then have two different workflows that start with a different screening assay. Again, I will also introduce this later. So we have the STX screening and the 0157 screening. And then furthermore, the identification of the remaining top 6 non-0157 STECs. I've mentioned the IMS beads for the isolation, and there is also an option, as I mentioned before as well, for the PCR confirmation from these IMS beads, which is a very nice tool that is used by many of our customers to confirm if they have something in their enrichment or not. The workflows are, of course, validated and certified. There is the NF validation, so AFNOR in Europe. I will not go into details for this validation. For this presentation, I will mainly focus on the AOAC PTM certification. And there are 3 features that I would like to introduce a little bit more in detail, which is the pre-razor treatment, which is an elimination of free DNA, and why this could be helpful for your lab. The UNG digestion, also another feature to reduce cross-contamination in a lab or to make the workflow safer. I think a very unique feature of our workflow. And then very briefly introduce the software solution that we have. The portfolio, as mentioned before, we have the BACGene E. coli 0157H7 kit. We have the BACGene multiplex for the STX green, which includes the STX1, STX2, and EAE genes. If you're interested in the separation of the STX, we have the 1 and 2 in different channels, so this can be identified as it is relevant for some of the countries. BACGene multiplex serotype kits then are made up of 2 different parts, and the identification of 3 O groups with each kit is possible. You can see the O groups here on the screen. The format of the kit is quite convenient, ready to use format. So we have in our PCR plates, the master mix pre-dispensed, and then the plate is sealed by a foil, as you can see in the lower right corner. So basically all you need to do is spin down, open, and add your lysate to the reaction well, and all the lysis reagents and the plastic for that are also, of course, supplied with the kits. There are different formats. We have 96 reactions for the screening kits, and 48 reactions for the serotyping kits, as these are normally not used on such a regular basis. Additional equipment is not a lot really that you need. You would need heating blocks for the lysis. There is a requirement for short centrifugation of the strips. So you would need a small lab centrifuge, pipettes, and then the thermocycler. Also, one nice part about this workflow, or in general our kits, I think, is that that we're using open systems, so you can basically run it on the Agilent ARIA MX and Biorad CFX instruments. These are the instruments that are certified, but you can also, of course, use these instruments for any other analysis that you're interested in. Our kits, the other kits that we have at Eurofins GeneScan Technologies, all run on these instruments, but also, of course, competitors will have lots of assays here, so you're not limited to a platform that runs only one company's assays. The workflow is quite an easy one. So we have an enrichment, as mentioned before, in MTSB in this case. For the AFNO, we have BPW, so this is why this is mentioned here as well, but we will focus on the MTSB. You transfer it into a sterile container, prepare your lysis buffer, which is basically just adding an enzyme to the lysis buffer and transferring it into the reaction wells. Then there is a short thermal lysis of 30 minutes, after which you need to spin down briefly. As I mentioned before, this is just to get the condensate out of the top of the lids, and then go into the PCR and run it on your instrument. After the PCR, as I said, we have the possibility to do the immunomagnetic separation, so the IMS. Of course, we do have all the relevant beads for this. As you can see here, there's not much needed in addition to this. There's a small rotator that you would need for the incubation of the IMS beads with your enrichment broth, and then you need a magnetic rag for the separation, and a vortex to get the beads in resuspension. So if we look over the workflow for this, when you come from the enrichment, basically you have a clarified enrichment that you mix with your IMS beads. You incubate those on the rotator for a certain period of time. You do the IMS separation. Then there are 3 washing steps after that, and then you can go into different detections. So here you can see that you could go on the plates as is described by the MLG, but as I said before, you can also then take these washed beads and go into PCR and basically confirm your results from the initial screening PCR on your beads, which is a further indication of the presence of an STEC. Here is an overview of the Latex kits that are also available through Eurofins Technologies. Don't want to go too much into detail with those because I think they're probably not that much needed in most of the labs. Now for the special features, and I would like to spend a little bit of time here. The BACGene pre-raser is something that we have certified with the kit, and there's always one question that comes up is, are all PCR reactions actually true positives or not? And the clear statement to that is, well, it's an indication that there is something there, definitely, because you will not have any false amplification, but it's not a statement that you actually have something alive there. So the answer has to be no, not really. So if we look at a scenario, why could it be that you get a signal but then you do not find any live bacteria? The easiest scenario that you have is basically if you think of a sanitation scenario. So you see here an overview of a bench. On the left side is before sanitation, and you see your happy bugs alive. Then you do the sanitation, and on the lower right part, basically you see the dead bacteria with their DNA. So if you take a swab on the left side, you will get a PCR signal after your enrichment, and you will get a cultural positive. So those two results will be in line. If you would do a swab on the lower right part, you would probably if you have a very high load of your bacteria on the surface, you would still get a PCR signal, maybe a very weak PCR signal, but you would get one because it's diluted in the enrichment broth. But culturally, you would not be able to find anything. So this is one of the reasons why sometimes people get unconfirmed results with a PCR. So what is our solution to that? It's basically the pre-RASER step, which is a free DNA removal. So if you look at the lower scenario, you would then basically digest your DNA and get a PCR negative that is then in line with the cultural confirmation. So a very nice tool, and that is also certified. In our workflow, as you can see here, it fits in right before the lysis, of course, because as soon as you lysed, then the DNA of the live bacteria is also free. So it's right before the lysis step, a short incubation step. And we have certified this for a broad range of bacteria. So you can use it for the Salmonella, Listeria, and also the STEC. Now the second one, and this is something that most people don't think of straight away. The last point was a contamination risk of live bacteria, basically in your sample, that are then killed off and leave their DNA in your material. There is also a second risk, especially in the STEC workflow, because you have to use PCR. I mean, there is no way that you can really find STEC just by cultural means, and PCR always involves the amplification of certain regions of the target genome. So you see on the top left corner a typical amplification curve, and you can see that on the left side it's very low, and then you come into a typical sigmoid shape of a PCR, and at the end you have a very high concentration. And what's that concentration? It's actually copies of your target DNA. So during the course of a PCR, you amplify the target DNA of interest in your sample to millionfold. So of course, this will normally never get into your lab because the PCR tubes stay closed, but accidents do happen from time to time. And I've been in technical support for some time before I joined Eurofins, and I have seen it more than once that there's actually a contamination due to an accident or due to really wrong handling of the tubes or whatever. So there is a risk that you actually contaminate your lab with these PCR amplicons and get a false positive result, as you can see on the left side. Now, what we do is, and this is quite a unique trick that is not used that often, and so I think it's a very unique feature of our assay. We label our PCR products. So when we amplify our DNA in the PCR for the detection, we label it. So you can differentiate between the genomic DNA and the PCR DNA. You don't need to do that. It's actually something that is done automatically, and it's just a very simple extra step that costs, I think, only 2 minutes in the whole PCR protocol on the cycler, and you don't even notice it, but there is an initial digestion of these PCR amplicons in your PCR reaction, and then only the DNA that is coming from a natural genomic source will be amplified in the subsequent PCR. So this is a very, very good contamination prevention that hopefully no one ever needs, but if you need it, it's extremely helpful to keep your lab running. So the next point is the FastFinder software. As I mentioned before, so this is our software to evaluate your runs. This is a software that can use run files from different cyclers. So, you know, we have the ARIA and the CFX, and it uses a very advanced machine learning algorithm. So it's quite good in training and identifying true amplification curves, which is much superior in comparison to the normal software that is used by the standard software that coming with the instruments. It can be integrated quite easily into the lab. So we have the different cyclers, and you basically just need to put in the raw data into that instrument, and it evaluates everything completely, and then you can export it as a PDF, as a report, or to go into LIMS as a CSV file or similar export files. Now coming to the next part, the workflows and the certifications. I've mentioned before that we have two screening approaches. The first screening approach is based basically on the historical fact that in the US, the regulation started with the 0157. And actually, even though the MLG has changed some time ago, and the FSIS opinion has changed to include the non-0157 STEC strains, a lot of the labs and customers still are actually using this workflow. So it's quite essential to have the workflow. I personally don't think it will last for very much longer because the direction is clearly going into the non-0157 screen as well. But this workflow would be starting with the 0157H7. If this is negative, you can report as a negative. We do have the option with our FastFinder software to evaluate with and without H7. So this is something also depending on the country, if it is required or not. The second step is then a confirmation PCR using the STX, the STX screen assay. If this is negative, then you can report it as a negative. If you have both of these PCRs positive, this would be a presumptive positive and going into the cultural confirmation. Now, the second workflow is the top 7 workflow. And here we basically go the other way around, as the MLG is doing as well. And actually, it was quite nice because we developed this workflow actually before the change of the MLG, of course, because that was like 2019. So it was right when we started our certifications. So it was quite nice to see that these official methods actually then took the turn into our direction as well. So the MLG and our workflow start with a screen. Here, again, we have the option in our software to choose, do we want to have only the STX being scored as positive, or do we want the combination of STX and EAE? So you have all the flexibility depending on the country that you want to export to make sure that you are compliant with the regulatory requirements. After that, we have the different serotype assays for the identification. If there's a positive coming up, if all these are negative, you can report them as negative, or you can go then into the cultural confirmation if you have a positive. Cultural confirmation, I only want to mention very briefly. You can see that there are different options here. You can just streak onto a plate and see if you can find it, or you can use the IMS beads, which I think is normally the best approach. Of course, I wanted to mention it here because you can see in this workflow then that you also have this optional PCR confirmation from the IMS beads. And as I mentioned before, we have a lot of customers who actually use that workflow at the moment, and they find it quite convenient if they don't go into the confirmation to actually double check on the IMS beads if they can confirm the results or not, to get more confidence in the results. And we found some, actually already quite a lot of STECs with this workflow then. So if we look into the protocols that are used, I think the most relevant matrices are for you the ground beef and the beef trim, which are very much on top here. So the 375 grams. And as I mentioned before, a very good dilution in 1 in 3 MTSB. So this is a very low dilution that we were able to certify, and which works very well. And also the incubation times, as I mentioned. So it's a very quick workflow with only 10 hours enrichment time. But as we have a lot of customers that require an overnight incubation, and we actually had some customers that said, well, we do need at least the 24, or not at least, but we need up to the 24 hours to make sure that we can actually process our samples in time. We made sure that we can actually go up to the 24 hours, which is also quite important to certify the longest period, because it's not always obvious that that a bacteria will still be alive after a very long incubation. It can be outcompeted by other bacteria in the enrichment broth and then maybe not be detected after the longest incubation anymore. Then we have, of course, the surfaces certified and the beef carcass cloth. I don't know if this one is used in Australia, but it is a USDA recommended tool for analyzing the carcasses. And then we have another product here as well, the flour and the romaine lettuce as relevant matrices. So in summary, coming to the end of the presentation, I hope it was informative for you, and I could show you the advantages of our workflow and get you interested. So I think the key points to take back from my point of view is that the field of the STEC detection is still really evolving. And I think most countries are not yet sure of where this will all end, basically. So the regulations will keep on changing, in my opinion. So having a method that is flexible and we believe that our system really meets the challenges very nicely. And as we've seen, we already have seen a change in regulation that the method could buffer very easily. It's due to its modular approach. It's quite flexible and efficient, as well as cost efficient, of course, to meet all the different regulatory requirements. Unique features I've mentioned. Just want to highlight again the certified free DNA removal, pre-raser, the UNG incorporation for cross-contamination prevention, and the non-proprietary enrichment media. So if a manufacturer cannot produce, you can go to the next manufacturer and are not limited to only one supplier. And it doesn't have any antibiotics. So MTSB is not the cheapest media, but still better than some of the antibiotic containing media, of course. And then the low sample to media ratios. So with this, I would like to finish the presentation and thank you very much for your attention. As I said, I hope it was informative, and if you have any additional questions, I will look into the questions in the chat now. But you can, of course, also later always ask me or my colleagues if there are any additional questions coming up. Yes, so there's one question that came in now, which is basically the definition, and I know this is always a big confusion, and this is also something that internally we discuss a lot, and you have to be very careful with that, the question between dilution and the ratio. So if we talk about a dilution, this is why I try to make it clear. Our dilution, it's a 1 in 3. So this means you're 375 grams in 750 milliliters of enrichment broth. So if that would be a ratio, then it would be 1 to 3. Then it would be 375 to the 1,100. What is it? 25. But in our case, yes, it's a dilution. So it's 1 in 3. So you need only 750 milliliters of enrichment broth. Thank you for that question.